when speed is low and then nucleus sediment first, and then when the rate of centrifugation increases, centrifugal force increases, then mitochondria they precipitate, and from microsomal fractions smaller elements are available, and the organelles can be separated, and they have all the common structure of cell membrane. Now we can discuss something about their anonymous topics. Some salient facts we have to discuss, and you can increase your knowledge from different books, internet, etc. Suppose the nucleus. Nucleus. It is present in almost all the cells, and this nucleus, they contains the chromosomes, and the chromosomes are the site of determination of the species and individual characteristics. Yeah. And chromosomes names come from chrome means color, colored substances. They are stained by the basic dyes. When you will stain the blood slide, then methylene blue will stain the nuclei of the WBCs. And the chromosomes, you all know, they are DNA, large DNA molecules, long DNA molecules, and they are compressed within that small space. Cell dimension you can remember, 10 to 20 μm in most cases, and nucleus is much more smaller. In spite of that, in which the DNA, the long thread-like structure, and it is accommodated. Accommodation is via the rounding around the bead-like structures. Bead-like, they are called nucleosomes. Suppose 2 meter long, a DNA chain is 2 meter long of a chromosome, and it is rounded against this histone type of protein structures. In this way, they are compressed, and they are rounding and rounding, and they contain DNA and the proteins, nucleosomes. 25 million nucleosomes within the nucleus, DNA chain, and they are double-stranded. You have in higher secondary texts, double-stranded, deoxyribonucleic, and RNA is single-stranded. Now, in this type, they are disposed 23 pairs. 23 pairs. One coming from the father and another coming from the mother. And in this way, they are resting cells, resting somatic cell, 46 chromosomes. And they bear the genetic material for the development of the species. And they contain the codes of protein synthesis, DNA to RNA, and from RNA the proteins are produced. Your higher secondary text have this type of discussion, even good discussion than a common text of medical profession. mRNA, messenger RNA, is produced from the DNA from one strand. Yeah. And they are there, and this process is called transcription. And it is produced within the nucleus. And it is like the beads, the histone proteins, and other proteins, and this is the DNA chains. And in this way they are, and in the resting cell, they are only the clumps of chromatin. chromosome and proteins chromatin, clumps of chromatin. But during cell division, they separate and they loosen from these beads, and only then their characters become clear under the microscope. Now DNA has a two strand, and then they separate, and from one strand, then this mRNA is produced. It is the coding strand, it is the template strand, and from which the mRNA is produced. It is single strand, and protein synthesis occurs within the cytosol, and this mRNA binds with the ribosomes. That is the ribosomes, and these ribosomes may remain free within the cytosol, or they may come in contact with the endoplasmic reticulum, where they form the rough surfaced endoplasmic reticulum. Yeah. And this, from this mRNA, proteins are synthesized, and there is the triplet code. Yeah. You can remember in your higher secondary course studies. Difference between the DNA RNA is the basically first is the RNA contains the ribose moiety, and it is called the deoxyribose moiety, and there is purine base and pyrimidine base. Purine base, that is the adenine and guanine present in the RNA and DNA, and in case of RNA there is uracil. In case of DNA, there is the thymine. Methyl uracil is the thymine. It is the basic difference. And there are hydrogen bonds in case of DNA, double stranded. Thymine to adenine, cytosine to guanine. In this way, the strands are. Suppose it is thymine, it is adenine, it is the guanine, and it is the cytosine. In this way, they are disposed. Hydrogen bonding. Detailed can you have any biochemistry text or even some physiology, they have only, I can remember only a few of them and can tell to you. That is very basic. From the, and this mRNA, so it is the DNA is double stranded, and their separation, it is the coding strand, and it is the template strand, TEMPLAT, ছাঁচ আর কি, ছাঁচের মধ্যে না হয় না, যারা সন্দেশ তৈরি করে দেখবে ছাঁচের মধ্যে ফেলে, একই রকম দেখতে একটা সারফেস হয়, টেমপ্লেট, and from this the mRNA is triplet code, that is the that is the nitrogenous base, 2 purine and 2 pyrimidine, and it is this, and from this triplet code, all the amino acids can be synthesized to united to one another to form the protein. Triplet code. Yeah. In case of human beings, it is, there is the, in mammals, AUG. It is the methionine. Yeah. Methionine. Triplet code means that is the 3 position. Any of this can take this place. If it is duplet code, is 4 into 4 is equal to 16. It is less than 20 that is present in the amino acids, so it is less. If it is 3, then 4 into 4 into 4, 64. And it is sufficient. And some are redundant, some may be stop codons. Yeah. Permutation, combination. So triplet code is sufficient. And it represents this, it is the methionine, and it begins with this. And there is whenever, and there is termination. Suppose UAA, UAG, and like, I cannot remember everything in details. When I am speaking from the mind, there may be some mistakes, and it requires thorough investigation so that I have committed a mistake. মানুষ তো একটা ভুল করার যন্ত্র একটা, কাজে আমার ভুল হয়, তাই তোমরা সেটা বই থেকে দেখবে, দেখে বলবে যে না আপনার এটা ভুল বলেছে, তাহলে তোমার নলেজটা পারফেক্ট হবে, বুঝতে পেরেছো? হ্যাঁ, তাহলে এটা কাজেই আমি সেই জন্য সাধারণত নোট বা এগুলো আমি পছন্দ করি না। And in this way it will proceed. And this, and suppose methionine, it will be carried by some elements, and it requires tRNA, transfer RNA, and enzyme that will this tRNA binds with it. Yeah. And there are enzymes for binding this with next amino acid. And in this case ribosomes, you have to know, these are the structures over which the proteins are synthesized. Their dimension is 22 into 32 nanometer, and they are produced from the information from nucleoli. The cells are growing, they have even more than one nucleoli, and they are the patches from which these ribosomes are produced. Ribosomes consist of RNAs and multiple proteins, and there are this type of structure, 40S and 60S structures, and over which this mRNA chains, these are produced, and from which the proteins will come out, beginning with mRNA. Clear? And those proteins that require secretion or transport to other part of the cell, they are produced within the lumen of the endoplasmic reticulum. Endoplasmic reticulum are structures, like they are hollow structures, tube-like structures connected to one another. And when these proteins are, suppose it is the protein that is going synthesis proteins, and it will enter into the endoplasmic reticulum. Endoplasmic reticulum like structure is, and they then combine with it. Some of the protein, most of the proteins required for secretion, as in secretion from the different glands, pancreas, salivary glands, or numerous other glands, endocrine or non-endocrine, and they require the secretion, and they are produced within the lumen of the endoplasmic reticulum. And when these ribosomes bind with it, they form at the cytoplasmic surface, they form this rough surfaced endoplasmic reticulum. Yeah. In some cases, secretion is not required, as is present in the hemoglobin within the RBCs, precursors of RBCs. Hemoglobin does not require secretion. Even presence of hemoglobin in excess amount in the blood in the free form is harmful. They can clog renal tubules, and they can produce other toxic effects. And in that case, they are produced within the ribosomes of the cytosol, but they are free ribosomes, not attached to any endoplasmic reticulum. They are free ribosomes. Yeah. But when there is secretion on this protein that is produced, ultimately it is to be transported to within the Golgi, within the cell membrane, and other structures, lysosomes, and in that case they are produced within the lumen of the endoplasmic. And whenever there is beginning of this protein synthesis, there is some signal peptide, hydrophobic type. They are formed of hydrophobic type of amino acids, and then they unite with the signal recognition particle, and then this translation stops, and then unite with this particular pore. There is a name for this, where they recognize this and they pore, and ultimately then they introduce this part, signal recognition particle, and they are then sectioned, and in this way this translation going on, and they end when they come into contact with this type of stop codons, UAA, UAG, or this like stop codon. When there is stop, and then they enter and they separate, and in this process going on, then there is the number of the ribosomes in the proteins. Then they form the polyribosomes. Yeah. Polyribosomes. Chain is growing, and there are these ribosomes at distances, and it's called polyribosomes. Rough surface endoplasmic. And in this way, these proteins are produced. And this within the endoplasmic reticulum, there are some primary modification of the protein chains. There is beginning of glycosylation. Preformed carbohydrate chains unite with it, and further modification is required. Yeah. And that is carried within the Golgi apparatus, another important organelle, Golgi apparatus. Besides this rough surfaced endoplasmic reticulum that bear the ribosomal units at their cytoplasmic surface, there are also smooth endoplasmic reticulum. There are no particles over the surface, they are smooth. They cannot synthesize protein, but they can synthesize steroid hormones, and they can detoxify some substances, and they are present enormously in the suprarenal cells, suprarenal cortex. Yeah. Lipid synthesis, steroid hormone synthesis, smooth endoplasmic reticulum. But protein synthesis occurs in the cytosol by the help of these ribosomes. Ribosomes are produced by the nucleus. They contain numerous proteins and also typical at least 2 ribonucleic acid, ribosomal RNAs. And they are this type of structures, 40S and 60S. And this mRNA, they contain this type of codes, and these codes are recognized by the tRNAs. tRNA bear this, their contain phosphate groups, and also enzyme are there for their activation. And in this way, one after another, by peptide chain is produced. Peptide chain, CONH group. In case of amino acid, this type of structure it is. And they combine to form CONH bond. Peptide. Accurate figure you will find in the text. This type of concept is required, and these peptide bonds are formed. and the tRNAs behave from when it is going from, it will change over the place among these ribosomes. ওই যে দুটো কাটা না হলে যেমন একদম করে না, দেখবে উল যখন বুনছে একটা কাটা ঢুকাচ্ছে, আরেকটা কাটা বার করছে, আবার একটা কাটা ঢুকাচ্ছে, একটা কাটা, এইভাবে চলছে, এইভাবে চলছে। মানুষের নেচারের মধ্যেই আছে, আর নেচারে যেই ফর্মগুলো এখানে আছে তারাও সেইভাবেই কাজ করছে। দুটো কাটা না হলে পরে সিনথেসিস হয় না, বুঝতে পারছেন? লংজিটিউডাল স্ট্রাকচার সেখানে তো একটা কাটা থেকে নিচ্ছে দেখবে, এবং তারা কথা বলছে, অনেক কিছু করছে, তার সঙ্গে সঙ্গে বুনে যাচ্ছে। কত সুন্দর সুন্দর সোয়েটার আগে লোকেরা তৈরি করতে পারত, এখন সেটা মেশিনে তৈরি হচ্ছে। এখন মহিলারা অন্যান্য বড় বড় কাজে যাচ্ছে এবং সেখানে বিভ্রাট ঘটাচ্ছে বহু রকম, কিন্তু কাজে এই যে কাজগুলো তাদের ছিল সেগুলো তাদের অনেকেই ভুলে গেছে। কিন্তু এটা নেচার এটা অনুমোদন করছে যে তোমার এখানে দুটো বিট না থাকলেও হবে না। Ribosomal RNA. Now Golgi apparatus. Golgi apparatus is present near the nucleus, and this endoplasmic reticulum has continuation with that of the nuclear membrane. In every cell there is single membrane, of course doubles. In case of nucleus, you will this type of membrane. This type of membrane. And from side this, so this is double membrane, and there are nuclear pores, and it is called perinuclear cistern, and it is connected with endoplasmic reticulum, rough surface endoplasmic reticulum. So there is continuation between them. বোঝা গেল? এখানে রাইবোসোম বসে রয়েছে, and this is the place for which the transfer of the larger molecules, nuclear pore complex. Otherwise, how can mRNA come through this, or ribosomal RNA can come through this nuclear pore, and proteins are transferred from outside to inside, or proteins are synthesized within this cytosol only, not within the nucleus. So mRNA will come from this rRNA, ribosomal RNA can come, even tRNA may come through this nuclear pore complex. And endoplasmic reticulum, rupture endoplasmic is continuation of the perinuclear cisterns, and these pore complex are large. They are formed of 8 subunits, and nearly 100 proteins are required. Pore complex, 8 subunits. Suppose you can draw a figure in your this type of structures. Numerous proteins are there. In this way, exact diameter I cannot remember, but through these proteins and mRNAs, they can come out, larger nuclear pore complex, because they are only permeable to very small molecules. So larger molecules require the permeability, and they are different types of proteins: importins, exportins, and exportins like this. And there are also other proteins for organization, and they are called RAN proteins. From biochemistry text, Harper text book, you can find the detail, and it is fascinating. And if you read carefully, from this point you can easily understand what is going on. And there is the active process, and there is the role of GTP and so. And I request everybody of you to consult this detailed movement through the nuclear pores from biochemistry text. Physiology text only mention this. Yeah. Now, what is occurring in the Golgi apparatus? Golgi apparatus, like dinner plates, stacks of the dinner plates, and Golgi apparatus near the nucleus, and they are close to endoplasmic reticulum. They are this type of structures. Six or so Golgi apparatus, and in which there is final formation of the proteins. Initial glycosylation occurs within the endoplasmic reticulum, and this protein then from this lumen, this protein is produced and it is coming into this by means of the vesicles, and this vesicle will bind with this, and so it is the cis form, cis site, and it is the trans site. And when this protein enters, partially glycosylated protein enters within yeast, then there is further glycosylations and further segmentation is required. It may be there are different types of enzymes. From one vesicle, and from this vesicle, and this go to in this vesicle. In this way, cis strands in this form, and ultimately from this the vesicle will come out, and they are the mature vesicle, and they are ticketed for different organs. Ticketed for different organs. Some for the cell membrane, some for the lysosomes and other structures. Even in the Golgi complex, they have different tickets, and they are different forms of the carbohydrate moieties, glycoproteins. Different type of glycoproteins that are produced. Yeah. And they are produced in this way that they can recognize the particular site of their insertion. যেমন তোমাদের যেখানে ট্রেনিং হচ্ছে, তোমরা যখন সার্টিফিকেট পেয়ে যাবে, হ্যাঁ, সেরকম যদি দেশের প্রয়োজন হয় তাহলে তোমাদেরকে ভিন্ন ভিন্ন জায়গায় পাঠানো হবে। সবাই তো সমান কাজের উপযুক্ত হবে না। দেখা যাবে কেউ কেউ এই ডিপার্টমেন্টের পক্ষে উপযুক্ত হবে, তাকে সেখানে পাঠাবে। সেগুলো হিসাব যেমন থাকে, এখানে তোমাদের যেমন হিসাবটা কম থাকে, পাস করে তারপরে MBBS ডিগ্রি দিয়ে ছেড়ে দিল, তারপর সে কি করবে না করবে কিছু জানে না। কিন্তু সেটা সব ক্ষেত্রে হয় না। যেমন সেনাবাহিনী যারা রিক্রুটেড হয় তারা ট্রেনিং হয়, ট্রেনিং হওয়ার পরে তারপরে বুঝতে পারে যে একটু দুর্বল প্রকৃতির, তাকে দিয়ে অন্য কাজ হবে। আবার যে খুব স্ট্রং অ্যান্ড স্টাউট, তারা হয়তো কমান্ডো বা ট্রেনিং পেল, তারা আরো ভালো জায়গায় পজিশন পেল। এরকম ভাবে তাদের ক্ষেত্র বিশেষ পাঠানো। এখানে সেই ব্যবস্থা করা আছে, ঠিক জায়গায় সে চলে যাচ্ছে। এবং ইট ইজ এন ইন্টারেস্টিং স্টোরি। দেয়ার আর স্নেয়ার প্রোটিনস, V snare proteins, T snare proteins, SNARE, snare proteins. Snare proteins, V snare, vesicle snare. It unites with the T snare, target snares. The final destination in this way. Soluble N-ethyl melamine attachment প্রোটিনের রিসেপ্টর, N-ethyl melamine attachment factor তার রিসেপ্টর, এটা হলো স্নেয়ার। স্নেয়ার only snare means there is this net. So this abbreviation is done so that we can commonly mean that this type of contain this type of net, and in this way it will recognize its corresponding part in a target organ. And there are also other proteins, and it is an active process, and it is an interesting part. In physiology test, you will find in detail. But in the biochemistry test, you can find how they occur. And whenever in this process of formation, the proteins that is destined for the lysosomes does not have the appropriate ticket, so that you cannot, lysosomal enzymes cannot enter within the lysosomes. They remains in the cytosol. And lysosomes will be filled by this material, it should degrade. Lysosomal storage diseases may develop, inclusion cell disease also. My knowledge regarding this is incomplete. I request you to go through this book and have the perfect knowledge regarding this. কারণ এটা প্রত্যেকের জানা আছে যে, এবং সবাই জানে যে এর বুদ্ধি কম আমার থেকে, সবাই বুদ্ধিমান। পৃথিবীর এখানে যত লোক চড়ে বেড়ায়, কাজে তোমরা এটা বইপত্র খুলে লাইব্রেরিতে দেখে নেবে। আই সেল ডিজিজ, ইনক্লুশন সেল ডিজিজ। So these are these organelles. Now mitochondria. We can discuss something about the mitochondria. Mitochondria. And these are the structures for, that is the generation of energy, you know, generation of energy. Aerobic process. Hundreds and thousands of mitochondria may be present in a cell. And their structure you can remember. There is double wall structures, outer lamella, this sausage type of structures, and the inner lamella is in this way. Their shape, sizes differ in cell to cell, but the basic structure is there. It is outside membrane and inner membrane, and these are the shelves or cristae. SHELVS, thak ache cristae, and it is the intermembrane space or intercrystal space, and it is the matrix space. And there are some structures like this within this. And in which this ATP is produced from ADP. And these enzymes present in this, and their permeability is variable. Outer membrane, its permeability is more. Molecules can easily traverse. But this inner membrane is not so. And there are some enzymes present within this space. And the other enzymes, they are all collected over this inner membrane. An electron microscope, this type of structures. Right? And they are the site of synthesis of ATP, adenosine triphosphate, from adenosine diphosphate, from the breakdown products of, that is the energy giving foods. Principal energy giving food is the carbohydrate. And this carbohydrate present in the plasma is glucose. Glucose enter into the cells, and within the cytosol it is broken down. To what extent it will be broken down? It is the, you have learned in the higher secondary text. It is broken down up to the level of the 6-carbon compound. Glucose is a 6-carbon compound, and it is broken up to the level of, and this process called glycolysis, cytosolic glycolysis. Emden Meyerhoff pathway. In your higher secondary text, they are already written. Emden Meyerhoff process. And it is broken down up to the level of the pyruvate, a 3-carbon compound. Pyruvate. CH3COCOH. And this pyruvate then enters within this mitochondria within the, and ultimately there is destruction of this within the aerobic process, and there are some enzymes are responsible for it, enzymes, and and that is the, and in this way there is this pathway, that is hydrogen ion accumulates within this space, This hydrogen ion then come through this complex and ATP is produced. There are 5 complexes of the inner membrane, and they are formed, complexes are formed mostly out of the proteins produced with the direction from the nuclear DNA. But there are 13 complexes that are derived from the mitochondrial DNA. And these and other features indicate that previously, billions years previously, it was a separate organism, and it enters into the eukaryotic cells. But its primitive character to some extent remains. It has its independent DNA genome, base pairs, 1, 6, 5, 6, 9 base pairs. Base pairs, BP, base pairs, circular base pair, and they have individual ribosomes, and within the mitochondria there is the scope of the formation of synthesis of proteins, and their coding is different from that of the mammalian codons. Okay? এবং এর সাহায্যে এবং আরো অন্যান্য প্রমাণের সাহায্যে বোঝা যায় it was a independent organism. It entered into a eukaryotic cells, mammalian cell, and it is helping in some way. It can also causes the death of the cells by apoptosis. Now, what are these general pattern that I can remember in this way? Suppose it is the mitochondrial outer membrane. So here there is the cytosol, in mitochondrial inner membrane, and it is the matrix space, and there are enzyme complexes 1, 2, 3, 4, 5. It is like this L pattern, inverted L pattern. This type of 3, 4, and it is the 5. Excuse me, it is the inner membrane, and it is the outer membrane. This is the outer membrane. And this is the cytosol matrix. And it is NADH dehydrogenase complex. Dehydrogenase complex. NADH dehydrogenase complex. And in this, there is the mitochondrial DNA and nuclear DNA. 7 proteins are from mitochondrial DNA and 39 proteins from nuclear DNA that is produced. They all come together to produce this complex. If you go through Harper's current text also, you will find in detail, and they are intelligible. But in physiology text, they explain in a simplified way. For the physiology, subject of physiology, some knowledge of biochemistry, previously it was a common subject. But there is extensive development of biochemistry, including its clinical application. So this subject has to be separated so that more intensive study is required. বুঝা গেল? আমরা যখন স্টুডেন্ট ছিলাম তখন একটা সাবজেক্টই ছিল, ফিজিওলজির ভিতরে ফিজিওলজি বায়োকেমিস্ট্রি একটা ছিল। পরে যখন প্রয়োজন বাড়লো তখন সেটা পৃথক হলো। অনেক জিনিস জানা গেছে, আগে পুরনো বই অনেক জিনিস জানা ছিল না। আর দিন যাচ্ছে, সেখানে সেখানে মানুষের বুদ্ধিও বেড়ে যাচ্ছে, কাজে আমাদের যে বোধ ক্ষমতা সেখান থেকে তোমাদের অনেক বেশি আছে। সেই জন্য তোমাদের জন্য সেপারেট সাবজেক্ট করে দেওয়া হয়েছে, যাতে অতিরিক্ত বুদ্ধিমান লোকেরা এটা শিখতে পারে। বুঝতে পারছো? যে এর জন্য প্রাচীন লোকদের যত তাড়াতাড়ি এরা ফিল্ড থেকে চলে যায় তত মঙ্গল। বুঝতে পেরেছো? তাহলে চাষবাসটা ভালো হয়। যেমন এখানে পুরনো গাছগুলো সব কেটে নতুন চারা লাগায়, নতুন গাছ হয়। সেই জন্য প্রত্যেকের পবিত্র কর্তব্য হচ্ছে আমাদের মতো লোককে তাড়াতাড়ি করে বিদায় করা। কিন্তু আমার যতটুকু সময়ের মধ্যে আমি তোমাদের এটা বলে দিচ্ছি, যাতে তোমাদের ইন্টারেস্টটা পাও। You should start yourself. And it is 39 proteins is from the nuclear DNA and mitochondrial DNA 7 proteins. And it is the succinate dehydrogenase complex, and it has no in this and in this case it is 4, and it is 1, 3 in this one. I may commit mistake, will find in the text accurate. Now what is the effect? Now it is ubiquinone cytochrome reductase, and then it is the ATP synthesis. Through 1, 3, 4, and this hydrogen ion chemiosmotic theory. It is called chemiosmotic theory. Hydrogen ion will come in this, and then hydrogen ion will come in this direction, hydrogen ion, and there is synthesis of ADP to ATP, and there are some transporters that can carry this ATP ultimately out of this mitochondria. Transport system, and different toxins or drugs can have effect upon the transport system of various components. So there are these complexes, 5 complexes: NADH dehydrogenase complex, succinate dehydrogenase complex, ubiquinone, cytochrome oxidase complex, and then cytochrome oxidase oxidoreductase complex, and then ATP synthesis. And from 1, 3, and 4, hydrogen ion will enter into this space, intermembrane space, and this hydrogen ion then enter through this space, and it is formed of different subunits. And when there is ATP formation, then there is some rotation through this, rotation of this part. They have different names, and they are formed of different subunits, and there are rotation. যেমন মাটির জিনিস যখন তৈরি করবে দেখবে একটা রোটেশন হয়, এই এটিপি সিনথেসিস সঙ্গে সঙ্গে তার একটা রোটেশন হচ্ছে, এনার্জি সেভাবে তৈরি হচ্ছে। It is oxidative process, and the carrier of the electrons, in this case it is ubiquinone or coenzyme Q in this case, and from this it is the cytochrome C from this level, electron carrier, and at the same time there is oxidation. Carbon dioxide is produced, and energy is there. Yeah. Biochemistry text describe in detail, but in physiology we have to know something about this. Electron carriers. It may be ubiquinone containing coenzyme Q or cytochrome C. It is the electron carrier. it unites with the oxygen to reduce it, and there is then from carbon compounds there is CO2 and H2 is produced, and at the same time the ATP is formed. And now in this way energy is produced. Energy is produced. From ADP and ATP is produced. In this. And the biochemistry text you will find even the number of these hydrogen ions by each unit. Somewhere it is 4, somewhere it is 2. And there are calculations of what is possible. From a glucose molecule by aerobic oxidation, how much amount of ATP can be produced. In older text, the 38 ATPs are produced by a glucose molecule. When it is completely broken down. Now, if this process is disturbed, and in that case the energy is consumed, but ATP is not produced. Oxidative phosphorylation. Oxidation along with phosphorylation, that is the generation of ATP. Suppose if there is some, due to some drugs or some defects, this hydrogen ion come out through this, not through this complex 5 or ATP synthase. Then there will be energy wasting. Yeah. But ATP is not produced. Disruption of oxidative phosphorylation. Dinitrophenol and other drugs. And it causes, there is this shunting path. So this hydrogen ion will not enter, may not go through completely through this. And so the energy production, energy is wasted, but ATP is not formed. ATP, it is a conserving process. Whenever there is ATP or any energy conserving process, some part is always lost. But if this is defective, then more is lost. বোঝা গেল? যেমন একটা হিটারের উপরে তুমি যদি তার যথাযথ মাপ অনুযায়ী বসাও, তাহলে অনেকটা হিটই সেখানে তোমার রান্নাতে কাজে লাগবে। কিন্তু তুমি যদি ছোট পাত্র বসাও, তাহলে সাইড দিয়ে লস হবে। তোমার এনার্জি কনজাম্পশন সেম হবে, কিন্তু তোমার বেনিফিটটা তুমি ওয়ার্কটা কম পাবে। And so it is no exception. Yeah. Now mitochondria is that it is within our us, the mitochondria is coming from the ovum. When there is fertilization, there is no chance of entry of mitochondria. There is a huge number of mitochondria within this sperms. What is the active movement? But whenever there is fusion of this, there is the sperm and ovum, then mitochondria has no chance to enter. So whatever mitochondria we get is due to, it is maternal origin. And mitochondrial 1, 6, 5, 6, 9 base pairs. It is responsible for 13 proteins. 13 proteins responsible. 2 other proteins are required for ribosomes of the mitochondria, and other for the tRNAs, 24 tRNAs and so on. Yeah. Now they have these, and in that case they have minimum repairing system. Whenever the DNA is produced in human being, from one DNA another DNA is produced in course of mitosis or second phase of meiosis. From DNA to DNA is produced, and so in somatic, if we consider it is a mitosis, and there may be defects, and there are many systems to detect these defects and repair. DNA repair system, and DNA repair system is poor in case of mitochondria. So mutations are much more than in comparison to that of the DNA produced by the nuclear genes. And some mutations are there, and they are of maternal origin. And they are present in those tissues where there is requirement of ATP is more, like muscles also, in nervous systems. there is a term MELAS, MELAS. Mitochondrial encephalopathy with lactic acidosis or different types of myopathies. In different texts or internet, you can search mitochondrial diseases. They are the list of diseases and their detailed elaboration. So initially, we have to go through this part in physiology text, and from internet and from library text, you can have much more information. কাজেই আমরা যেটা বলছি সেটা খুবই অল্প পরিমাণ আমার জ্ঞান বুদ্ধিতে যা কোলায় সেটা আমি বলছি, হ্যাঁ, এবং এটা প্রয়োজনের তুলনায় খুবই নগণ্য। তোমরা যখন ওগুলো পড়বে তোমাদের জ্ঞান বৃদ্ধি পাবে। Now so it is mitochondria. There is another compound that is called peroxisomes. Peroxisomes. Microsomal fraction. It is present in the microsomal fraction. When centrifugal speed or the gravitational force is maximum, and then this particles is sediment. Whenever this rotational speed is low, then initially there is deposition of the precipitation of the nuclei, and it is to some extent more, then the precipitation of mitochondria or endoplasmic reticulum, and when it is maximum, and then from the supernatant of earlier part, then there is separation of this ultracentrifugation, and it is called microsomal fraction, and this peroxisome is present. Peroxisomes, and their name come from this, they can generate peroxide. They have oxidase enzymes that can generate H2O2, and also they contain catalase that causes a breakdown of this hydrogen peroxide, and catalase is one of the marker of this organelle, along with uric acid oxidase, marker of this organelle, 0.5 μm in diameter, and there are specific proteins over it by means of which it can get its proteins introduced. Peroxisomes, these proteins synthesized elsewhere within the cytosol, not from endoplasmic reticulum, and it will enter, and for this there are some unique signal sequences called peroxines, and there are some particular, that is the proteins in this membrane of the peroxisome which will allow it within this, and in this way numerous enzymes enter into it. About 40 enzymes are within this, and these enzymes along with the enzymes in the cytosol responsible for the metabolic processes like long chain fatty acids, bile acids also, this type of, and in this process this is produced, and there are some disorders that are related to the entry of this defect in the entry of this enzymes also. Adrenoleukodystrophy, Zellweger syndrome, Zellweger syndrome. Zellweger syndrome. Zellweger syndrome. Adrenoleukodystrophy. Even fatal disease can be produced. Some may be mild, and similarly in different biochemistry texts and from the internet you have this, you have the detailed discussion of this. There are signs, symptoms also, and it is expected that you utilize your phone to have some information regarding this. Some drugs that has been shown to affect this proliferation of these peroxisomes. Whatever may be the way, they can cause proliferation by acting over the nuclear genes. PPAR they are called. Peroxisomal proliferation activator receptors, they within the nucleus. Similar type of receptors are present for the steroid hormones, vitamin D and other substances. that can bind with the DNA, and some of these preparations, some of the drugs, and they are alpha, beta, and gamma. Thiazolidine dione is a compound that acts upon this gamma type of receptor, PPR, and in this way it can be used. It increases insulin sensitivity. So it is the drug present in the market. I do not know whether they are used nowadays. They have some toxicity. And this, on the other hand, there are some chemical agents or drugs, they can have the effect of alpha on beta. And in case of alpha, then there is this, not in case of gamma, that is the energy conservation, and it is energy utilization. PPAR binding drugs that causes similar to they unite with the appropriate receptors for them, and they may be used biochemically. And Zellweger syndrome is a almost lethal disease, and you should find out in the internet to have it, how, in where it can have their effect. Now, lysosomes we have discussed. Lysosomes. Time is short. Lysosome, they are these membrane bound structures. It may be of different types of irregular shape also, lysosomes. And within this there are some proteins that can digest any type of enzymes. They can destroy any types of material that is available. And they are within the lysosomal fluid, the pH is low, 5.0 or like. And these enzymes are hydrolyses. As they are active in the acid medium, they are acid hydrolyses. And there is pump, sodium hydrogen pump, that can direct this from low concentration cytosol to within the lysosomes. The benefit is that if there is some leaking occurs of the enzymes, and within the cytosols where the pH is not favorable, it is not immediately active. And these lysosomal enzymes are produced from the Golgi bodies, and there are certain tickets by means of which it will enter the lysosomes. And whenever there is some genetic defect in these enzymes, then what will occur? There is some substrates over which these enzymes, and they will accumulate within this. This is called lysosomal storage diseases. Lysosomal storage diseases that will accumulate. There will be enlargement in the liver, spleen, and there is erosion of bones and different types of defects. They are rare diseases, but their existence and their analysis helps to explore what is going on in the body. Lysosomal storage diseases like Fabry's disease, suppose alpha glucosidase deficiency, it's called Fabry's disease. Complex carbohydrate, and it is called Fabry's disease. Similarly, there is Gaucher's disease. Tay-Sachs disease. And it may cause different defects including blindness, mental retardation, and different types. They act upon different enzymes. And in this case, Gaucher's disease is the beta glucocerebrosidase. And in this case, it is the amino sugar splitting enzyme is absent in this case, Tay-Sachs disease. There are also other diseases, Niemann-Pick diseases, and in the biochemistry text you can have a chart like this. There is the over which carbohydrate at which point it will act. Lysosomal storage diseases. Yeah. So time is almost over. So I stop now. Next day we will discuss about the cytoskeleton and along cytoskeleton, and it contains, there is these different types of fibers and also molecular motors, and if time is available to some cell adhesion molecules and also apoptosis. Next day we will discuss, and in this way I will try to partially complete this general discussion, and more important part, that is the body fluid, the cell membrane, membrane potential, that are more important. And you study the books recommended unofficially for common students are Debashish Pramanik. You go this book and you learn at least the basic part that you need to know.